Once upon a time, there was a very brave and generous girl named Alex, and she decided she wanted to help other kids with cancer, and thousands of other volunteers, thousands of you, helped her do it. Tonight, I have the honor and privilege to tell a couple of stories about the difference all of you are making for the kids that we all hope want to serve. So my story begins a little bit outside Detroit, where I was born and raised. I did my MD PhD at the University of Michigan, my residency in WashU in St. Louis, and then came to Johns Hopkins and the National Institutes of Health to do my fellowship in pediatric oncology. My research has been in the laboratory of Elias Zambitis, another pediatric oncologist who is also a stem cell engineer. In 2014, I had the privilege of being promoted from fellow to faculty at Johns Hopkins. As many of you are familiar, in this age of very tight NIH funding, that jump from fellow to faculty is extraordinarily difficult. Into that funding gap has stepped a number of foundations, ALSF leading them. Thanks to the ALSF, they made possible my chance to become a member of the faculty at Johns Hopkins and the chance to continue my research. In fact, almost 1 out of every 3 professors at Hopkins today are ALSF awardees. Investigators like my friends Brian or Stacy, who like me had the chance to join the faculty because of the generosity of all of you. Or senior investigators who have been able to start brand new projects in revolutionary new fields because of the funding the ALSF made possible. What I'd like to do is to spend a couple of moments telling some stories of the actual kids that the foundation has been able to help through the application of science to the development of new cures. This is Grace. Grace is not her actual name, of course, but her family has very generously let us tell her story. Many years ago, when I was a first-year fellow, she was a wonderful little girl running around her grandparents' farm on vacation, chasing her baby sister, chasing the butterflies, petting the animals, and spending a wonderful summer until she got sick. And then she got really sick. She had a really sharp country doc who was able to ask the right questions, do the right tests, and realize that her body was being consumed by leukemia. She was so sick that her life was ebbing away just in front of their eyes. It was actually one of my first nights manning the phone at Johns Hopkins. And I remember that night, working with that country doc, calling for emergency transport, getting her to the pediatric intensive care unit where doctors fought to save her life. She ended up needing a breathing tube. She ended up needing life support. She ended up needing neurosurgery to correct the tumor bursting into her brain so hard it nearly came out of her skull. But after a lot of hard work and many, many touch-and-go days, she was stable enough to return home to Johns Hopkins, where we continued chemotherapy. And her T-cell leukemia was under control for a couple of months. Came back. We immediately launched a second line of chemotherapy, new agents directed specifically against the T-cell leukemia. After hard work on her part, we were able to get that back into control and then give her a bone marrow transplant from her matched sister to replace her sick marrow with her sister's matched own. And that kept the leukemia away for 6 months. Then the T-cell leukemia came back. So she had been through intensive chemotherapy, and her T-cell leukemia came back. She had been through a perfect match bone marrow transplant from her sister, and the leukemia came back. 10 years ago, we would not have had anything else to offer. But this is Professor Simons, one of my mentors at Johns Hopkins, and herself, many years ago, an ALSF awardee. Her career begun thanks to a Young Investigator Award just like mine. And the work that she did then, and work that she continued to do with other colleagues at Johns Hopkins, with other scientists and colleagues all across the country, made possible a new generation of bone marrow transplants called haploidentical bone marrow transplants. Let's take a moment to talk about that. Bone marrow transplants traditionally are done from matched siblings or matched donors, strangers, volunteers, an immune system that matches yours immunologically. There are disadvantages to that, however. The problem is that the reason why you got cancer in the first place was in part because the tumor figured out how to hide from your immune system. In you or me, whenever a cancer cell appears, our normal immune system cleans it up. That's why we don't get cancer a dozen times a day. When a tumor figures out how to trick your immune system, that's when it begins to spread. That's when it begins to grow. That's when it takes over. And so you give chemo, you can knock it down, the tumor comes back. You can do a bone marrow transplant with a matched immune system, but the tumor already figured out how to hide from you. It figures out pretty easily how to hide from the match. However, if you can figure out how to do a bone marrow transplant from someone who only half matches you, all of a sudden the tricks that the tumor used to hide from your immune system and hide from the matched transplant don't work anymore on that transplant that only half matches you. All those ways to hide don't work anymore. In addition, when you're using a half-match transplant, the number of donors rises dramatically. After all, every child is half their mother, half their father. If you can do a transplant from a half-match, that means every kid who has a parent potentially has a donor, and that's huge. So many of you heard the desperate searches for donors, the attempts to try to find someone out there who matches, the drives that people generously donate to so that maybe they can be the stranger that full matches. Well, if you do a half match, that gives so many more kids the life-saving chance. So all those things give you a new option. But when it came Grace's turn, nobody had ever done a haploidentical match with booster shots to try to fight down a T-cell leukemia that had already come back multiple times. We did a literature search. We talked to colleagues of ours all over the world, and people said, it seems like a really good idea. The logic, the science, it all makes sense, but nobody's ever done it. And there's side effects and there's challenges, but on the other hand, we were out of options. And so Grace and her family bravely decided to try. They felt that even if it didn't work, what we could learn from the experience might be able to help other kids moving forward. And so almost 3 years ago, she underwent the haploidentical transplant with donor lymphocyte infusion booster shots to try to fight off her recurrent T-cell leukemia. And this is Grace today. It's been 3 years. Ever since she left the hospital after her first transplant, she's only been in the hospital 4 additional days, 2 each for each of her booster shots. She's been back to school. She's been a big sister. She's been a ballerina. She comes to camp every summer and we run around together. She loves butterflies and Disney princesses, and she's still trying to decide what she wants to do when she grows up. But that's exactly what she's going to get, the chance to grow up. And all that because many, many years ago, the ALSF began making a series of investments in faculty members and in research that ultimately paid off. Thanks to the foundation, we had the opportunity to present her story to the world. We wrote a scientific paper, and our team includes 3 investigators, myself, Professor Simons, and Professor Schaefer, who all were supported by the foundation, and the work itself likewise. Now, when other doctors and other families face the same crisis that Grace's family did, they will find something. They will find a report, and they will have an option that could work for them as it did for Grace. But it's not just blood tumors that the ALSF has made a huge difference in individual patients of mine. This is Adam. Adam is now a running around rambunctious 4th grader who is now 4 years out from having a tumor that filled nearly half of his brain. On the left is Dr. Eric Rabb, professor. When I was a fellow, he was a professor who helped me take care of Adam. Eric originally also was a young investigator. He was looking at ways to try to crack open DIPG, to try to look at different aspects of the human genome in ways that people haven't looked at before. And he made some amazing discoveries that he is now advancing thanks to a second, larger grant from the ALSF. With those awards, he will hopefully be able to take those discoveries that he's made and turn them into cures. But together, we've been able to make a difference, us together, for kids like Adam as well. So for all the kids we've been able to help, there are still too many kids we can't. How are we going to solve that? Well, we're going to try to solve that by taking the most revolutionary discoveries in human science and seeing if they can apply to the care of kids. That's the basic science that the ALSF has enabled me to pursue. The idea is to use adult stem cells, adult pluripotent stem cells, to try to model pediatric cancers. Well, what does that actually involve? Let's talk about that. The specific cancer that I'm pursuing is retinoblastoma. Retinoblastoma is one of the most common cancers of the eye, and it's one of those tumors that actually appears before birth. As the pediatrician, you go into the nursery, new babies, you count the toes, you count the fingers, and you check the eyes. Most of the time, you get a red reflex. You see the back of the retinas, you get those nice, reassuring red dots. But once in a very infrequent while, you don't see the red, you see white. You see white because the inside of the eye is filled with white tumor, retinoblastoma. It used to be uniformly fatal. There's been a lot of work to try to improve that. Now kids who have retinoblastoma, they go through chemotherapy, they go through radiation, they have laser therapy, cryotherapy. Many of them end up losing one eye. Others lose both. And even after all that, many do not survive. And the ones who even do survive, the therapy that they got gave them more cancer. Clearly, we need better answers. A lot of research has helped us understand more about where retinoblastoma comes from. All of you will remember from high school and from college, where do we come from? We come from a sperm and an egg form a cell called the pluripotent stem cell. That one cell has the ability to become the rest of us, become hair, eyes, skin, heart, and some of them become retinas. So from that original pluripotent stem cell, some of them become eye stem cells and then go through a series of steps and become a normal retina. We have learned that in retinoblastoma, something goes wrong with one particular gene called the RB1 gene. And when that mistake happens, something goes off the rails. That process that goes from the normal stem cell to the final retina breaks down, and instead of getting a beautiful retina with the ability to see and life in all its beautiful colors, you get this cancer. The question is, where? Where in this long drive from the original stem cell to the tumor, where does the process break down? If you can figure out where it broke down, you can figure out what happens there, you can make a drug for it. Not that simple. The problem is, as I mentioned, retinoblastoma is one of those tumors that happen when you're born with it, which means that you can't watch it develop because, well, it was happening inside mother. You could perhaps watch it in mice, but it turns out mice don't actually get retinoblastoma the same way we do. For whatever reason, their biology is different enough that you don't get the same thing. So you can't watch it happen in the patients. You can't watch it happen in mice. But what if? What if there was some way we could watch it happen in a dish? What if we could actually make that happen inside, underneath a microscope, watch it day by day, and figure out where does it go wrong? And that's where next generation technology comes in. So it turns out that you can actually take a skin cell, a blood cell, take a piece of hair off and those little cells at the bottom there, you can actually turn them backwards into that very first original cell. You can take an adult cell and turn it backwards into that original pluripotent stem cell called an iPS cell, and then turn them forward again into anything you want: heart, neurons, skin. That technology lets you basically take a patient's cells, turn them back into that pluripotent stem cell, turn them forward into anything you want. And that can do a lot of amazing things. For example, you could take a patient's skin cells, turn them back to an adult stem cell, and turn them forward into blood to help kids with blood cancers. Or many of these kids go through terrible amounts of chemo and their hearts get damaged. You could take a patient's blood cells, turn them back to an adult stem cell, turn them forward into new heart tissue, and help repair their hearts. And together with colleagues of ours at the Wilmer Eye Institute, my mentor, Elias Zambitis, was able to take blood cells, turn them back into adult stem cells, and turn them forward into retinas. We can take this process, we can make retinas in a little dish, you can shine light on them, and they make little electrical impulses, just like real retinas do. So if you can make a normal retina from an adult stem cell, then maybe you can model the cancerous stem cells into retinoblastoma. And that is the project that the ALSF has provided me seed money to begin to pursue. Basically, we're working to turn the normal adult stem cell, introduce the mutation we know causes retinoblastoma, And then together with our colleagues at Wilmer, actually watch that whole process grow up. And basically, we're going to hope to see where retinoblastoma comes from. There are a lot of technical challenges, a lot of issues. With any new technology, there's a lot of groundwork that goes into it. But the foundation set here will provide the beginnings of many years of work, which, just like for Grace, will someday hopefully give us new answers. And the impact goes well beyond just retinoblastoma. Yes, we will develop new cures for that, but there are other tumors in kids and adults that involve the same gene. So if you learn something in retinoblastoma, it'll help lots of other kids with osteosarcomas, and it'll help adults too. Science is science. You learn a fact, it applies everywhere. And beyond that, the ability to engineer stem cells, the science that we gain from this process, will help us build other tissues to help other problems in other kids with cancer, as well as many other diseases. Again, science is science. And so the impact of all the support we get to fund one little thing ripples and ripples and ripples. And that leads to the kind of final point. People ask all the time, you know, what can 1 cup of lemonade do? A lot. Biology is a science where every little step adds forward, and each little step is really quite cheap. A single dish, little 6-well dish of stem cells, costs $3.90. That's only a couple of cups of lemonade. A single immunofluorescence assay, another fundamental experiment, $9. That's a couple of cups of lemonade. A few cups of lemonade funds an experiment. A couple of those experiments become a data point. A couple of those data points become a figure. A couple of figures become a paper. A couple of papers becomes a cure. Every one of those cups of lemonade is one step up the mountain. Each step by itself doesn't look like much, but every single step gets you closer to the summit. That's how we're going to beat pediatric cancer. That's how 1 cup at a time you beat pediatric cancer, because you climb Mount Everest 1 step at a time, but you never go backwards. And so when people ask, what do those pins buy? What do those ties buy? What do those cups buy? It buys a fact. You add up enough facts, you get a cure. We've done it before, we'll do it again, thanks to all of you. And that leads to my last slide. I've told you stories about patients, but the last story is mine and my family's. This is my cousin Roy. 40 years ago, he was diagnosed with a brain tumor, and he was one of the first generation of kids to successfully survive. His life was saved by an oncologist at the University of Michigan, the same oncologist who 30 years later will be my first teachers in pediatric oncology. The doctors who saved his life, who helped my family, I was inspired as a little boy to become a pediatric oncologist so that I could help other kids the same way that those doctors helped us. And that was all made possible because of many generous mentors, colleagues, and all of you who helped support my research, my training, and the opportunities that I had to make a difference for others. I'm just one investigator. There are thousands like me over the many years the foundation has been part of. And everything that we have done, everything we will do, is because of all of your generosity. You, us, all of us together, we will work until every cancer has a cure, until every kid can go home. And that's what we're here about, to fight and win this battle together. Thank you.